Welcome back to Med Studious, and welcome to one of the most clinically important and frequently examined topics in general pathology, cellular hypertrophy. If you look at the image on screen, you can see a cell shown in two states. On the left, a baseline normal cell with sparse internal contents. And on the right, the same cell after hypertrophy, filled with more mitochondria, more structural proteins, more endoplasmic reticulum, more of everything. That visual difference tells the entire story. Hypertrophy is not a disease in itself. It is a cellular strategy. It is the cell's way of saying, the demands on me have increased, and I am going to build myself up to meet them. In this video, we are going to go far deeper than the definition. We are going to understand exactly what triggers hypertrophy, how the cell senses the need to grow, what molecular machinery drives that growth, how physiological hypertrophy differs from pathological hypertrophy, and what the real-world clinical consequences are. By the end, you will be able to answer any exam question on hypertrophy with complete confidence. And more importantly, you will be able to apply this knowledge when you are standing in front of a patient. Let's begin.
Every story of hypertrophy begins with the same trigger, an increase in workload that disrupts the cell's existing state of homeostasis. Remember, homeostasis is the balanced equilibrium state in which the cell efficiently performs all its required functions. Think of it as a perfectly calibrated scale, balanced, stable, and sustainable. Now, imagine placing a heavy weight on one side of that scale. The balance is lost. The cell is now under stress, being asked to do more than it is currently equipped to handle. This is precisely what happens when you begin an intense exercise program, for example. The sudden mechanical load placed on muscle cells is far greater than what those cells were previously managing at baseline. The cell's existing protein content, enzyme levels, and energy-generating capacity are all insufficient for the new demand. This mismatch between supply and demand is the fundamental driver of hypertrophy. And here is the critical insight. The cell's response to this disruption is not passive. It is an active, intelligent, precisely orchestrated program of internal restructuring. The cell does not simply struggle under the load. It detects the stress, signals its nucleus, activates specific growth genes, synthesizes new proteins, and physically enlarges itself to restore balance. This is cellular adaptation through hypertrophy, purposeful, measurable, and life-preserving.
When a cell faces increased demand, it has two fundamental strategies available, and understanding the difference between them is essential. The first strategy is hypertrophy. The cell increases its own individual size. It produces more enzymes to catalyze reactions faster, more structural and contractile proteins to do more mechanical work, and expands its overall functional surface area so that more biochemical reactions can occur simultaneously. The cell essentially upgrades itself from a compact car into a high-performance machine. Same vehicle, massively more powerful. The second strategy is hyperplasia. Instead of making one cell bigger, the tissue creates more cells. The workload is distributed across a greater number of identical workers, each operating at a manageable individual capacity. Think of it as hiring more staff rather than overloading one employee. Both strategies achieve the same ultimate goal, matching the cell's collective functional output to the new environmental demand, but they use completely different biological mechanisms to get there. Hypertrophy requires increased protein synthesis within existing cells. Hyperplasia requires cell division and proliferation. And critically, as we will see in a moment, not all cells have the option to choose between these two strategies. Some are permanently locked into one path, and that distinction has profound consequences for how certain organs respond to disease.
Here is one of the most important and most frequently examined rules in the entire pathology of cellular adaptation. A cell's ability to undergo hypertrophy versus hyperplasia is determined entirely by whether or not that cell is capable of dividing. Non-dividing cells, cells that have permanently exited the cell cycle and lost the ability to replicate, have only one adaptive option available to them when stressed: hypertrophy. they cannot make more copies of themselves. So instead, they grow larger. The classic example is the cardiac myocyte, the muscle cell of the heart. Cardiac muscle cells are terminally differentiated. They do not divide in adult life. When the heart is placed under increased pressure, as in hypertension, the individual cardiac muscle cells have no choice but to enlarge. There is no other path. This single biological fact is why hypertension, left untreated, leads to a massively enlarged, thickened heart, because the only adaptation available to cardiac cells is hypertrophy. Dividing cells, by contrast, like the smooth muscle cells of the uterus, retain the ability to both enlarge and multiply. They have dual adaptive options, hypertrophy and hyperplasia. This rule of division is not just academic theory. It is the mechanistic explanation behind some of the most important cardiac conditions in clinical medicine. Memorize it.
Now that we understand what hypertrophy is and when it occurs, we must make a critical distinction that runs through all of pathology: the difference between physiological and pathological hypertrophy. Physiological hypertrophy occurs as part of normal, healthy body functioning. There is no underlying disease. There is no abnormality. The cell is simply responding intelligently and appropriately to legitimate, normal physiological demands. The hypertrophy is purposeful, proportionate, and fully reversible when the stimulus is removed. The athlete's enlarged skeletal muscles are the textbook example: pure physiological hypertrophy. Pathological hypertrophy, by contrast, is disease-driven. The stimulus is not a healthy physiological demand, but an underlying abnormality, a disease process that forces cells to enlarge not because growth is appropriate, but because the cell is struggling under a pathological burden it was never designed to carry. The enlargement is a compensatory response to something going wrong, not something going right. And crucially, while pathological hypertrophy may initially be adaptive and helpful, it often eventually becomes harmful, reaching a biological ceiling beyond which the enlarged organ begins to fail. This physiological versus pathological distinction is fundamental. Every time you encounter hypertrophy clinically or in an exam, your first question must always be, is this a normal response or is something wrong? The answer changes the diagnosis, the prognosis, and the treatment entirely.
In the absence of underlying disease, physiological hypertrophy is driven by three primary categories of trigger, and understanding all three gives you a complete picture of how healthy adaptation works. The first trigger is mechanical workload. When a cell is physically stretched, compressed, or subjected to repetitive mechanical force, as in exercise, physical labor, or the pressure changes of pregnancy, the physical deformation of the cell membrane is itself detected as a stress signal. The classic example is skeletal muscle hypertrophy from resistance training, where mechanical tension on muscle fibers directly activates growth signaling pathways. The second trigger is hormones. Chemical messengers circulating in the bloodstream can directly instruct cells to grow. Estrogen, for example, drives the massive hypertrophy and hyperplasia of uterine smooth muscle during pregnancy. Testosterone drives skeletal muscle hypertrophy. Growth hormone drives widespread tissue growth in developing organisms. The third trigger is growth factors. Locally secreted signaling proteins such as insulin-like growth factor 1, also known as IGF-1, and transforming growth factor beta, or TGF-beta. These molecules are released by neighboring cells in response to mechanical or chemical stress, and they bind to specific receptors on the target cell's surface to initiate the growth program. Together, these three trigger categories cover virtually every scenario of physiological hypertrophy you will encounter.
Let us make this concrete with a real, tangible case study. Look at the cross-section of a skeletal muscle fiber on screen. On the left is the baseline state, a muscle fiber with a modest diameter and relatively sparse internal organelle content. On the right, after just 6 months of consistent progressive resistance exercise, the same fiber is dramatically larger. The individual muscle fibers have expanded their diameter significantly. Inside each fiber, there is now more contractile protein, specifically more actin and myosin filaments. More mitochondria have been produced to generate greater quantities of ATP to fuel the increased contractile work. More sarcoplasmic reticulum has developed to handle the enhanced calcium signaling required for faster and stronger muscle contractions. This is workload-induced skeletal muscle hypertrophy in its purest form, a direct, proportionate, and fully physiological response to a sustained increase in mechanical demand. And here is the clinically important property of this type of hypertrophy. It is completely reversible. When the mechanical stimulus is removed, when the person stops exercising, the cells no longer receive the signal to maintain their enlarged state. Protein degradation begins to outpace protein synthesis, and the muscle fibers gradually shrink back towards their original baseline size. This reversibility is the hallmark of physiological adaptation. Remove the stress, and the cell returns to normal. This is the fundamental principle that separates adaptation from permanent pathological change.
Our second case study takes us into the remarkable world of reproductive physiology, specifically the hormonal hypertrophy of the uterus during pregnancy. Pregnancy is a normal physiological state, not a disease. The uterus must accommodate a growing organism that will increase in size by several orders of magnitude over 9 months. The primary hormonal driver of this extraordinary expansion is estrogen. Rising estrogen levels during pregnancy circulate through the bloodstream and bind to specific estrogen receptors on the surface and within the smooth muscle cells of the myometrium, the muscular wall of the uterus. This hormone receptor binding directly activates the cell's internal growth program. The smooth muscle cells, called myometrial cells or leiomyocytes, undergo dramatic hypertrophy, each individual cell increasing enormously in length and volume. Simultaneously, estrogen also promotes hyperplasia in these cells, since uterine smooth muscle cells retain the capacity to divide. The result is a uterus that transforms from a small, pear-sized organ, weighing approximately 60 to 70 grams in the non-pregnant state, into a massive structure capable of housing a full-term baby, increasing its capacity by a factor of roughly 500 to 1,000 times. After delivery, as estrogen levels fall, the hypertrophic and hyperplastic stimulus is removed, and the uterus undergoes involution, returning to its pre-pregnancy size within approximately 6 weeks. A perfect demonstration of hormonally driven, fully reversible physiological hypertrophy.
Now we move from the healthy to the harmful. Pathological hypertrophy occurs when cells are driven to enlarge not by normal physiological demands, but by a persistent underlying disease state that places the cell under abnormal, unrelenting stress. Look at the pressure gauge on screen. It is deep in the red zone. This is the cellular environment in pathological hypertrophy, a cell continuously subjected to excessive, disease-generated pressure that it was never designed to withstand. Unlike physiological hypertrophy, where the trigger is appropriate and the growth is proportionate, pathological hypertrophy is the cell's desperate attempt to compensate for an abnormality it cannot resolve. And here is the critical difference in outcome. While physiological hypertrophy enhances organ function and is fully reversible, pathological hypertrophy initially maintains organ function but progressively compromises it. Over time, the pathologically hypertrophied organ becomes structurally abnormal. Its architecture is distorted. Its blood supply becomes relatively inadequate for its enlarged mass. And eventually, despite or even because of all that hypertrophic growth, the organ begins to fail. Pathological hypertrophy is simultaneously the cell's attempt to save itself and, paradoxically, one of the mechanisms by which it ultimately destroys itself. The most important clinical example of this, and the most heavily examined, is cardiac hypertrophy driven by hypertension.
Look carefully at the 2 hearts on screen. The heart on the left is normal. Its walls are of appropriate thickness, its chambers have adequate volume, and its structure allows it to efficiently fill with blood during diastole and forcefully eject blood during systole. The heart on the right belongs to a patient with long-standing, uncontrolled hypertension, high blood pressure. Because the left ventricle must pump blood against a chronically elevated arterial pressure, it faces a dramatically increased pressure workload with every single heartbeat. Since cardiac myocytes cannot divide, their only adaptive response is hypertrophy. They grow larger, packing in more contractile proteins to generate more force. Initially, this hypertrophy is compensatory and actually maintains cardiac output. But look at what happens to the structure. As the ventricular wall thickens enormously, the internal cavity of the left ventricle becomes smaller and stiffer. The heart now struggles to fill properly. Additionally, the hypertrophied muscle mass outgrows its own blood supply. The coronary arteries that were adequate for a normal-sized heart are now insufficient for the massively enlarged muscle mass. This creates relative ischemia within the heart muscle itself. The result is diastolic dysfunction, reduced cardiac output, and eventually congestive heart failure. This is pathological hypertrophy at its most clinically devastating, an adaptation that begins as compensation and ends as catastrophe.
Let us now consolidate everything into a high-yield diagnostic comparison matrix, exactly the kind of structured thinking you need for both exams and clinical practice. First, origin. Physiological hypertrophy originates from normal body functioning. Pathological hypertrophy originates from an underlying disease or abnormality. Second, primary triggers. Physiological triggers are mechanical workload, hormones, and growth factors operating within normal physiology. The primary pathological trigger we focus on is chronic high blood pressure, hypertension, though other pathological states like valvular heart disease also apply. Third, key examples. The gold standard examples of physiological hypertrophy are skeletal muscle enlargement from exercise and uterine enlargement during pregnancy. The gold standard example of pathological hypertrophy is cardiac muscle enlargement from hypertension. Fourth, mechanism and outcome. Physiological hypertrophy is a reversible adaptation. Remove the stimulus and the cell returns to baseline. Pathological hypertrophy, while initially compensatory, leads to progressive structural compromise of the organ and eventually to functional failure if the underlying disease is not treated. Drill this matrix into your memory. In an OSCE, in a written exam, or in a clinical discussion, being able to rapidly compare physiological and pathological hypertrophy across these 4 dimensions will demonstrate the depth of understanding that separates an excellent student from an average one.
Now we go deeper into the molecular machinery of hypertrophy. How does the cell actually sense that it needs to grow? The answer begins at the cell membrane, which functions as a sophisticated sensory trigger board, continuously monitoring the cell's external environment for stress signals. There are 3 key receptor systems involved. The first is mechanical sensors, specifically integrin proteins. Integrins span the cell membrane and physically connect the extracellular matrix outside the cell to the cytoskeleton inside. When the cell is subjected to mechanical stress, pressure, stretch, or shear force, this physical deformation is transmitted through the integrins directly into the cell's interior, where it is converted into biochemical signaling cascades. The second sensor system is hormone receptors, specifically receptors for angiotensin II and estrogen. Angiotensin II is particularly important in cardiac hypertrophy. Its binding to AT1 receptors on cardiac muscle cells is a major driver of pathological cardiac growth, which is precisely why ACE inhibitors and angiotensin receptor blockers are used clinically to prevent and reverse cardiac hypertrophy in hypertensive patients. The third system is growth factor receptors, including receptors for TGF-beta, which when activated trigger receptor phosphorylation and initiate downstream signaling cascades. Hypertrophy always begins at the perimeter. The cell cannot decide to grow until it has first received and processed the external signal.
Once the cell membrane receptors have been activated by mechanical stress, hormones, or growth factors, the next stage of hypertrophy is signal transduction, the process by which the external surface signal is translated into an internal biochemical command that reaches the nucleus. The 2 most important signal transduction pathways involved in cellular hypertrophy are the PI3K pathway and the AKT pathway, and they typically work together in sequence. Here is how the circuit works. The activated receptor on the cell surface triggers the enzyme phosphoinositide 3 kinase, better known as PI3K. PI3K then phosphorylates and activates the serine threonine kinase AKT, also known as protein kinase B. Once activated, AKT acts as a master regulatory switch, initiating a cascade of downstream effects that collectively promote protein synthesis, inhibit protein degradation, and drive cell growth. Specifically, AKT activates mTOR, the mechanistic target of rapamycin, which is the key regulator of protein synthesis in the cell. mTOR activation accelerates ribosomal activity and increases the rate of translation of growth-related mRNAs into new proteins. This PI3K-AKT-mTOR axis is also highly relevant clinically. It is one of the most targeted pathways in cancer pharmacology, with drugs like rapamycin and its analogs used to inhibit uncontrolled cell growth. Understanding this pathway is therefore not just important for pathology, it is foundational for pharmacology as well.
Once the PI3K-AKT signal cascade reaches the nucleus, something remarkable happens. Something that reveals just how deeply the cell's growth program is embedded in its genetic history. The arriving biochemical signals activate a group of transcription factors, proteins that bind directly to DNA and switch specific genes on or off. The three most important transcription factors in cardiac hypertrophy are GATA4, MEF2, and NFAT. These transcription factors do not activate brand new genes that the cell has never used before. Instead, they reactivate a specific set of genes that were active during fetal development, but were switched off after birth, the so-called fetal gene program. These dormant embryonic genes are awakened, like sleeping soldiers being called back to duty. Once reactivated, these fetal genes drive the transcription of mRNA, which is then translated by ribosomes into two critical categories of new proteins: contractile proteins and enzymes, including heavy chains of myosin and actin, which increase the cell's mechanical power, and structural proteins, which expand the cell's physical architecture and increase its size. This reawakening of fetal genes is clinically significant because the fetal isoforms of contractile proteins, such as beta-myosin heavy chain, are actually less efficient than their adult counterparts. This contributes to the eventual contractile dysfunction seen in pathological cardiac hypertrophy. The cell tries to grow its way out of trouble, but the proteins it produces in the process are functionally inferior.
And now we arrive at the final step, where all the molecular events we have traced, from membrane receptor activation, through signal transduction, through nuclear gene activation, manifest as a visible, measurable, macroscopic change in the organ. The pathway flows in three clearly defined levels. At the microscopic level, the activated fetal gene program produces a massive surge of newly synthesized proteins: contractile proteins, structural proteins, enzymes. These proteins accumulate inside the cell, filling it with increased functional and structural content. At the cellular level, this protein accumulation causes the cell to physically expand. Its volume increases, its surface area grows, and its functional capacity rises. Multiply this expansion across the millions or billions of cells that make up an organ, and the effect becomes visible to the naked eye. At the macroscopic level, the level of the whole organ, the result is a heavier, structurally denser, stress-resistant organ. In physiologic hypertrophy, this produces an organ that functions better under the new demands. In pathologic hypertrophy, it produces an organ that is structurally compromised, architecturally distorted, and heading toward failure. This three-level cascade, from synthesized proteins to expanded cell to heavier organ, is the complete story of hypertrophy, told from the molecular to the macroscopic. And it is a story that bridges basic science and clinical medicine in the most elegant possible way. Thank you for watching Med Studious. If this lecture helped you, please like, comment, share, and subscribe. Your support keeps this channel growing.